ion-dependent co-transporter system is used. In this system, hydrogen-sodium exchange system is used which established a proton gradient. This gradient is used to transport the dipeptides and tripeptides. Established a proton gradient meaning due to the action of this pump, the concentration of hydrogen ion was higher in the intestinal lumen than in the intestinal cell. At the same time, the sodium-potassium ATPase pump brings the sodium to the outside. This pump, the sodium-hydrogen exchange protein, it brings sodium inside the cell. For every 3 sodium that is pumped outside, 1 sodium is brought inside by this channel that is present on the apical side or luminal side. This pump also pumps hydrogen ion to the outside of the cell. So the work of this sodium hydrogen exchange protein is to exchange 1 sodium for 1 hydrogen. So due to this action of this pump, it allows the concentration of hydrogen to be higher in the lumen than in the cell. There is another membrane protein, and that is hydrogen ion dependent co-transporter that allows passage of hydrogen ion into the cell down its concentration gradient. This gradient has been established as a result of sodium-potassium ATPase and sodium-hydrogen exchange pump. The di and tripeptides also travel with these hydrogen ions. Intracellular peptides act on di and tripeptides to break them into constituent amino acids. The intracellular peptides act on di and tripeptides to break them into constituent amino acids. These amino acids then enter the bloodstream either by passive diffusion or by co-transporters. This is the explanation for the absorption of di and tripeptides. So just look at the marks before answering. If they've asked only to make the diagram, then you make only the figure with all the labels properly. You should label all the pumps properly. Okay, this is another method, another technique for the absorption of the amino acids. Another process. This is known as gamma glutamyl cycle. This process is not that important as compared to the previous one, but just for your knowledge. So this cycle, it occurs in 3 main locations in the body. It occurs in the small intestine, in the kidneys, and in the liver. The main function, but for now we are concerned about the occurrence of the cycle in the small intestine. The main function of the cycle is to maximize the absorption of amino acids from the extracellular space. For this case, we want to maximize the absorption of amino acid from the intestinal lumen. So let's say that this is the intestinal lumen where the products of protein digestion, the amino acids are present. First, we have the glutathione inside the cell. So do you remember what is glutathione? How is glutathione made? Glutathione is made up of glutamine, cysteine, and glycine. So this process, the gamma glutamyl cycle, is started with the glutathione molecule. This glutathione molecule, it goes to the surface of the cell, and here it interacts with this specific enzyme, glutamyl transferase. So glutathione is inside of the cell, it goes to the cell surface. where it interacts with this enzyme glutamyl transferase. This enzyme is a transporter or transmembrane protein. What it does is it cleaves off the glutamate from the glutathione. So you know that glutathione is made up of glutamine, cysteine, and glycine. So glutamine is getting cleaved off from cysteine and glycine. The glutamate is then it gets stuck to the cell surface. Glutamate is then transported to the extracellular space still attached to this enzyme. This glutamate is then able to interact with the free amino acids that are present in the extracellular space and bind them. This will then form gamma glutamyl amino acid on the extracellular site. Then it will get transported into the cell. So this gamma glutamyl uh, amino acid is getting into the cell. So in order to utilize this amino acid, it is cleaved off from the glutamate and it is utilized. The glutamate is finally regenerated into glutathione through several steps costing a total of 3 ATPs. 1, 2 and 3 ATPs are used to again make a molecule of glutathione. So firstly, we had glutathione, which is made up of three amino acids. Cysteine and glycine are separated. Glutamate, it interacts with the enzyme present on the cell membrane, glutamate transferase. It goes to the extracellular space. It binds one amino acid, brings it in, and then the amino acid is released. And again, glutathione is regenerated in series of steps which utilize 1, 2, 3 ATP. So by looking at this trigger, you can say that when the body utilizes this process for the absorption of amino acid, it is very expensive. You need 3 ATPs to bring in one individual amino acid inside the cell. So for this reason, this gamma glutamyl cycle is not a common transport system for amino acid. So if you're asked about the absorption of amino acid, you can easily write the previous one, sodium dependent cotransporter system. Only if you are specifically asked to describe this gamma glutamyl cycle, you write this one. But if you are only asked to write absorption of amino acid, you can simply write about sodium dependent cotransport system. This gamma glutamyl cycle, it occurs in three main locations in the body: the small intestine, the kidneys, and the liver. The main function of the cycle is to maximize the absorption of amino acids from extracellular space. Glutathione is made up of glutamate, cysteine, and glycine. This is the explanation. Glutathione molecule goes to the surface of the cell, interacts with gamma glutamyl peptidase or glutamyl transferase. Glutamate gets cleaved off from cysteine and glycine. Glutamate is then transported to the extracellular site. Glutamate interacts with free amino acid within the extracellular space and binds them, forms gamma glutamyl amino acid on the extracellular site, gets transported into the cells. In order to utilize this amino acid, this is cleaved to release free amino acid and glutamate. Glutamate is finally regenerated into glutathione through several steps costing a total of 3 ATPs. This cycle is responsible for taking up the amino acid from urine in the kidneys so that amino acids are not lost in the urine. In the small intestine, we are concerned about this for now. In the small intestine, this cycle makes sure to take up as many amino acids as possible. So as I said, the products of protein digestion, amino acids, dipeptides and tripeptides are only absorbed by our intestinal cells. But there is an exception to this. For a short period immediately after birth, the small intestine of infants can absorb proteins and polypeptides as well. So this uptake of proteins and polypeptides, it occurs by the process of endocytosis, but this process is there only for a short period after birth. The importance of this is uh, there is transfer of maternal immunoglobulins to the offspring. You've heard that breast milk is the best for the baby, right? So why do you think that is? The breast milk immediately after birth, they contain the immunoglobulins, which are intact proteins. So through the breast milk, these immunoglobulins are transported to the infant. So uh, the offsprings, the babies, they can take up the maternal immunoglobulin because their intestine is able to absorb the intact proteins and polypeptides for a short period after birth. So this is the importance of this process. There are some abnormalities of protein digestion and amino acid absorption. So you can see that for proper digestion to take place, you need the pancreatic enzyme. So if these enzymes are not there or deficient, or if there is some disease of the pancreas, then it leads to abnormalities of protein digestion. So there can be different causes like chronic pancreatitis, cystic fibrosis, or surgical removal of pancreas. In these conditions, it causes loss of undigested protein in the feces. Next condition is Hartnup disease. This is an autosomal recessive disease. It affects the absorption of non-polar amino acids, mainly tryptophan. So what is Hartnup disease? It is autosomal recessive. Transport defect of tryptophan is seen. Neutral amino acid transport defect. I hope you remember what neutral amino acids are. And in this condition, it causes pellagra-like lesions, which is characterized by 3Ds: diarrhea, dementia, and dermatitis. Gardner disease is an autosomal recessive disorder caused by impaired neutral amino acid transport in the mush border membrane of the small intestine and the proximal tubule of the kidney. So when tryptophan is defective, there is deficiency of NAD, NADP, which are the coenzymes of niacin, and also serotonin and melatonin, because tryptophan can be converted to serotonin, melatonin, and niacin. Niacin is a precursor to nicotinamide or necessary component of NAD. We have studied about this in the vitamins. So this is one of the defect of protein digestion and absorption, the Hartnup disease. And another is protein intolerance. Many food proteins can act as antigens in the human. When they act as antigens, they trigger immune response. So mainly the cow's milk protein, soy protein, and egg protein, they lead to intolerance. The clinical features include loose stool, which may contain blood, abdominal cramps, coughing or wheezing, runny nose, watery eyes, itchy skin rash, often around the mouth, swelling of the lips, etc. So you've also studied about lactose intolerance. Lactose intolerance, now what was happening was after consumption of milk and milk products, you're getting symptoms like loose stool, abdominal cramps, right? So it is almost the same here. If you take cow's milk, then you see the features of protein intolerance, which also comprises of loose stools, abdominal cramps. But Lactose intolerance is not an allergic condition, whereas protein intolerance is an allergic condition. That is why you see the responses such as coughing, runny nose, watery eyes. Allergic feature is audible in case of protein intolerance, which is not seen in case of lactose intolerance. So this is the mechanism. Why is protein intolerance occurring? Because normally the di and tripeptides which enter the brush border of mucosal cells, they are immediately hydrolyzed into single amino acids and are then transported in the blood. We talked about this during the absorption of di and tripeptides. But sometimes, in rare cases, large molecules may pass paracellularly between the epithelial cells and enter the bloodstream. So these are immunogenic in nature and cause antibody reaction leading to food allergy. So this is the mechanism of protein intolerance. So there is one more enzyme that is there in protein digestion, renin. You please find out about this. It is found in the stomach of infants and children, but is absent in adults. So this completes our protein digestion. Now moving on to digestion of the dietary lipids. So now we will talk about the next nutrient that gets digested, which are the fats. The most common form in which we consume fat is triacylglycerol. So 90% of our dietary fat is triacylglycerol. The remaining consists of cholesterol, cholesterol esters, phospholipids, and free fatty acids. In the first year, you've studied about cholesterol, you've studied about cholesterol esters, and phospholipids as well. So you know how triacylglycerol is made, what is it composed of, everything. So this is our dietary fat composition: 90% triacylglycerol, and the remaining other lipids: cholesterol, cholesterol esters, phospholipids, and free fatty acids. So same as in case of protein, fat digestion is not there in the mouth because the saliva does not have any lipase. The digestion of lipids, it begins when the food reaches the stomach and it is catalyzed by the enzyme lingual lipase. Lingual lipase originates from the glands at the back of the tongue. And there is also another enzyme, the gastric lipase, which is made by the gastric mucosa. So this lingual lipase, it acts on the triacylglycerol molecules. Those contain fatty acids of short or medium chain length. So normally triglyceride contain 16, 18 or 20 carbons, but these lingual lipase, they only act on the triglyceride that have short or medium chain length fatty acids. How many fatty acids does one molecule of triacylglycerol have? It is called 3 fatty acids. Glycerol esterified to 3 fatty acids is triacylglycerol. Normally, there are long chain fatty acids which are esterified to glycerol to make triacylglycerol, but this lingual lipase, it only acts on triacylglycerol molecules which have fatty acids of short or medium chain length. These same triacylglycerol having short or medium chain length are again degraded by gastric lipase, which are acid stable enzymes. So the triacylglycerol molecule is containing short or medium chain length. They are mainly found in milk fat. This is the significance of lingual and gastric lipases. They play an important role in the digestion of neonates since milk is the main source of energy. I said that these lipases, the lingual and gastric lipase, they act on triacylglycerol containing short and medium chain fatty acids. So these kind of triacylglycerol are found in milk. So that is why these are important for digestion in the neonates. These also play vital role in digestion of lipids in pancreatic insufficiency such as cystic fibrosis and other pancreatic disorders. So the main enzymes that help in the digestion of lipids are present in the pancreatic secretion. But if the pancreas are defective, if there is some disease like cystic fibrosis going on, if the pancreas is not making all those enzymes, then even the work of these lingual and gastric lipases will be appreciated because some amount of digestion is going on and the diet is likely modified. So this is the digestion of lipids in the mouth, in the stomach by lingual and gastric lipase. So this is it for today. In the next class, we'll complete about the digestion of the dietary lipids, and then we'll talk about its absorption as well.